Okay, I think I'm going to get going. So, welcome everybody to this. I think it's the fifth one in the series now, the second semester series of our H3D Foundation webinar series. I'm Nicola Elliott Wong. I'm the program coordinator at H3D Foundation, and it's our great honor today to host Dr. Lee Graves. I'm going to read his bio. He's going to be speaking to us about the application of affinity proteomics to study the pharmacology of drug action today. Dr. Lee Graves has been a professor in the Department of Pharmacology at the University of North Carolina, Chapel Hill since 1995. Dr. Graves obtained a PhD in biochemistry at the University of Illinois at Champaign-Urbana, studying protein degradation. He then performed postdoctoral studies in the lab of Dr. Edwin G. Krebs, the University of Washington, studying the regulation of eukaryotic protein kinases. The focus of the Graves lab has been to apply proteomics to elucidate the pharmacology of drug action with a focus on kinase-mediated cell signaling. His lab has been successful at identifying new kinase substrates, feedback responses, mechanisms of cancer drug resistance, and regulation of metabolism. A second major area has been studying small molecule activators of the mitochondrial protease ClpP. Using an immobilized pharmacophore, his lab was the first to discover the mitochondrial protease ClpP as the target for ONC201 and the related anti-cancer compounds. We're really honored today to have you, Dr. Graves. I'm going to hand straight over to you. And for everyone listening, please enjoy. As usual, questions, there'll be a Q&A at the end. So please take notes as you go.
Okay, thank you. I hope everybody can hear me all right. I really appreciate this opportunity to talk about some of our research. This is an area that we've focused on for almost 30 years now. And like Nicola said, I'm going to start off talking about some of our kinase studies. And then at the end, I'm going to talk about some of the more recent stuff that we've been working on. Okay, so all our work really is rooted in proteomics. We have a fantastic team at UNC that supports all our research, state-of-the-art instruments, allowing us to look into the proteome at a very deep level and to understand basic elements of protein expression changes, protein phosphorylation changes, key things that really regulate a lot of biology. So today what I'm going to talk about is some of our affinity-based proteomics studies to look at kinases and to study what we call the kinome. The second third of my talk, I'm going to talk about a new project where we use affinity-based proteomics to discover an unknown drug target. Okay, well, to start with, a little history. So in the beginning, there was really only one kinase, and that was discovered by these two guys, Ed Krebs and Ed Fisher. Now, they were awarded the Nobel Prize in 1992 for this work, actually when I was in the lab, but the work was done back in the 50s. And at the time, this idea of phosphorylation as a regulatory mechanism was not at all understood. In fact, when their work first came out, there was a lot of questions of whether or not this was really how things work. But they were fortunate enough to have hot radioactive ATP, and they showed that the very first protein kinase was active in converting phosphorylase from an inactive state to an active state. Since that discovery, this field has exploded, and we now know that in the human kinome, there's approximately 500 kinases of varying flavors, different substrate specificities, different structures. In general, as we'll see, the structure of kinases is highly conserved throughout nature. What's interesting is that this is a very large enzyme family, and that many of these enzymes are mapped to disease loci or cancer amplicons, and so they're attractive targets for the pharmaceutical industry. And another reason that kinases have been so popular over the last 30 years is that they're very highly druggable. We now can design molecules that at nanomolar concentration have very high effective inhibition of protein kinases. And that's because of some beautiful work that came out of Susan Taylor and other people's group, particularly Susan's group, where they published the very first structure of the protein kinase known as the cyclic AMP-dependent protein kinase. And what that showed us was how both ATP and peptide substrates bound. And once we understood better how ATP bound to this interesting bilobal structure as shown here, and here's our ATP binding in the N-terminal part, then we could design inhibitors to prevent ATP from binding. And that gave birth to this entire field of kinase inhibitor design and application. The most famous compound being what's known as imatinib or Gleevec, which was the first to be used to show that you could inhibit a kinase that was associated with the disease. In this case, it was the BCR-ABL kinase and chronic myelogenous leukemia. And that this kinase inhibitor would bind very tightly into the ATP pocket and prevent ATP from binding, thereby turning off the kinase and stopping the disease. And since then, many other kinase inhibitors have been designed that are selective for their targets. Gefitinib for the EGFR, and I think I have abemaciclib, which is a CDK4/6 inhibitor shown over here. Now, there are approximately 80 FDA-approved kinase inhibitors that are in clinical use, many that have emerged not just for cancer, but for other skin diseases and other potentially important diseases. Okay, so kinase inhibitors have a place, as I mentioned, but kinase inhibitors also have limitations. And what's shown in this slide is a very well illustrated example of that. BRAF, when it becomes mutated, is often the driver of melanoma, and this has been well established. Single point mutation in BRAF turns it into a hyperactive kinase. Not surprisingly, people discovered that if you design an inhibitor such as vemurafenib that blocks BRAF, we can block the growth of these melanoma. Well, this patient here on the left has serious melanoma, underwent treatment with vemurafenib and showed promising changes in the beginning. After 15 weeks, he showed what looked like a complete loss of cancer. But by 23 weeks, the cancer had returned and the inhibitor no longer worked. And so one of the big challenges with kinase inhibitors is discovering why they lose efficacy and how can we circumvent that. And there are a couple of well-known ways that kinase inhibitors or kinases can become resistant to inhibitors. One, is shown in the upper right-hand corner here, is that the kinase actually mutates. And in the example here is shown what we call the gatekeeper mutation, where the kinase has now inserted an isoleucine in place of threonine. And this is the BCR-ABL kinase I mentioned earlier. And now the drug no longer binds into the pocket. It's excluded from the ATP pocket, and ATP binds, and the kinase becomes active again. Now, what choice do we have to deal with something like that? Well, in this case, we have to go back to the bench and design a molecule that avoids this gatekeeper mutation. And that's what's been done in chronic myelogenous leukemia. Second and third generation inhibitors have been designed that will now inhibit the gatekeeper mutation. A second and also important form of resistance to kinase inhibitors is upregulation of what we might call a surrogate kinase. For example, in the EGFR case, inhibitors inhibit the EGF receptor like gefitinib that I mentioned. However, upregulation of kinases like MET, which is another receptor tyrosine kinase, can circumvent the action of the inhibitor on EGFR and begin to phosphorylate substrates that the EGFR would phosphorylate, thereby bypassing the action of this drug. So it's critical that we understand what kinases are being upregulated or what kinases are changing in a way that we can now target MET in combination with the EGFR. Okay, so other reasons to study the kinome. What we've learned over the years is that the kinome is a highly interconnected network, that one kinase regulates another. There's feedback regulation, there's crosstalk. There are a number of examples now that we can understand that if we inhibit one kinase, we lead to the activation of another kinase. or that another kinase, like the surrogate kinase I just mentioned, becomes upregulated. So I think it's important to emphasize that the kinome is dynamic. And I like this example from physics. For every action, there is an equal and opposite reaction, Newton's third law. And so in essence, this applies to the adaptation, or what we'll call kinome reprogramming, in response to kinase inhibitors. Okay, so traditionally, how have we studied the kinome? Well, for a long time, we focused on the phosphoproteomics end of things. How does the network of phosphorylation events change in response to changes in the kinome? And we'll talk about the limitations of that in a second. A more recently developed approach was developed by a company known as Kinative, which they basically have used an affinity probe to bind to active kinases to assess which ones are now active under different conditions. And then the third approach I'm going to talk about is what we call kinobeads or MIBs, which is basically an affinity enrichment approach, which we can use to look at how kinases change under different conditions. Okay, so please stop me if you have questions. Otherwise, we'll keep on rolling through this. I hope my slides are clear to everyone. Okay, and most successful is we can apply more of these approaches in combination. So first, phosphoproteomics. This has become a very effective way to look at what substrates are changing. And the mass spectrometers have gotten so much better, our methods have gotten so much better, that we really can dive deeply into the phosphoproteome and see how different phosphorylation sites on proteins are changing. The challenges are, it requires that we deconvolute these thousands of phosphopeptides and try to determine which kinase put that phosphate group on which substrate. And there's a lot of redundancy. Some kinases will phosphorylate multiple substrates. Some substrates are phosphorylated by multiple kinases. And then some of these sites are not well represented in the phosphoproteome and are difficult to study by our traditional methods. So in essence, it becomes a bit like the old fairy tale Humpty Dumpty fell off the wall. We're trying to put these pieces back together and infer who Humpty Dumpty actually was before he broke into all these pieces. So the second approach is one that I mentioned, known as kinative. And kinative, again, relied on the knowledge that this is how kinases bind ATP. Having the crystal structure from Taylor's lab really allowed us to see that. And to see that there was a lysine residue that was in close proximity to ATP when the kinase was active. So by putting a biotin group on there, you could basically tag the active kinases and then use streptavidin to pull the kinase back out, degrade the kinase, look at the kinase peptides and determine which kinases were active. So this is a very clever way that people could look at kinase activation. And it does work, but some of the limitation is that there's again kinase redundancy, like the Sark family kinases are very, very similar. So tagging this active site won't tell you which member of the family it is necessarily. And that's sometimes one of the challenges, is to figure out, again, which of these kinases is truly active. And the third approach, which we're very popular or very fond of, I should say, actually was evolution of ideas, starting with immobilized ATP from Hasted's group at Duke. And then a few European groups discovered, well, if we put kinase inhibitors on a Sephiros bead, we can catch kinases with an affinity better than we can with ATP. And we expanded that to show that we could put multiple kinase inhibitors on there to capture a broad swath of the kinome from any tissue sample that we wanted to, cell lines, whatever we wanted to study in a new way. And what's shown at the bottom here is just how certain kinase inhibitors are more selective, like BisX binds a smaller number of kinases, but unique ones. And then compounds like VI and PP58 bind well over 100 kinases. And we have some other ones now that we use that bind up to 200 kinases. So the idea is that we want to get as global an analysis of the kinome in a single snapshot. And then coupling this with quantitative mass spectrometry, we can say, did this kinase go up or did this kinase go down under these conditions? What can we learn about how the kinome as a whole is changing in response to this? And I have to point out that all this work was done in collaboration with Gary Johnson here at UNC. He and I worked to build the system, and in his lab has really developed a ton of really exciting results as a result of this, as well as ours has. Okay, so I'm just going to go through a couple examples. We won't have time to go through everything, obviously, of how we use these multiplex inhibitor beads, or MIBs, to study kinome adaptations. In particular, we'll talk about cancer examples, since that's been one of the most popular applications. More recently, we've been focusing with a couple of groups on studying how viruses affect the host kinome and whether or not that gives us opportunities to target proteins in response to viral infection. And then lastly, which has been really popular in our group, is how do we define kinase inhibitor specificity? Can we use this approach to discover new targets for kinase inhibitors that weren't known? And I'll go through a few examples of that. Okay, so the first example that I want to go through is one that's been well referenced now, a very exciting study that came out of Gary Johnson's lab that I was part of. And it really started as a simple question. We know that this pathway, starting from a receptor like the EGF receptor, activates the MAP kinase pathway. And so MAP kinase pathway is often activated in cancers, including breast cancer. Could we apply inhibitors to this pathway and understand something about the kinome? So we use a well-defined MEK inhibitor, which blocks this pathway, and ask what changes. And this paper, like I said, was published in Cell. The lead author was James Duncan, a very talented postdoc then. He now has his own lab at Fox Chase, where he's doing kinome reprogramming studies using MIBs and other technologies. Okay, so what did we learn from this? Well, like I said, we started off very simply asking what happens to the kinome if we inhibit one kinase, that is MEK. And surprisingly, in addition to effects that we saw in MEK as we'd expected here, so just to point out that kinases below center line were all displaced or downregulated after treatment, where kinases to the top of the center line were all upregulated after treatment. And this was a short treatment, 24-hour treatment of a breast cancer cell line with the MEK inhibitor, as I mentioned. We discovered these cells are quickly developing in resistance to kinase inhibitors. And we looked more closely, it turned out it was a family of kinases. Oops, sorry, went the wrong way. And the family of kinases are part of this, what we call the receptor tyrosine kinase family. It's like the EGFR that I mentioned earlier. And this was both a surprise and a concern. In general, we don't want to be upregulating tyrosine kinases because they drive the proliferation of the cell. And so this gave us an example that the kinome was reprogramming or remodeling in response to this. And so we used this approach then to look more deeply at what's happening and whether or not there could be a therapeutic advantage to this knowledge. And to make a long story short, the discovery came down to an important finding that c-MYC protein, when MEK was inhibited, became inactive. And when c-MYC, when it is not phosphorylated by ERK, it becomes degraded, as you can see over here. There's a loss of MYC protein. Well, when MYC protein is lost, cells upregulate receptor tyrosine kinases, as can be shown in the upper right here, such as PDGF, VEGF, strongly upregulated as a result of loss of MYC. And if you block the degradation of MYC with bortezomib, which is shown here, you don't see this upregulation. So a really key point here was that this reprogramming was transcriptional in nature, and that it had to do with the loss of phosphorylation of MYC, which allowed the de-repression of these kinases, which were in essence there to reactivate cell growth. So the question was, could we then combine this with an inhibitor that blocked RTKs like sorafenib and see a therapeutic advantage now that we knew that this kinome reprogramming was happening. And this experiment here showed very nicely that here's the mice tumors growing. Sorafenib by itself has very little effect, but the combination of sorafenib plus the MEK inhibitor caused a much greater tumor regression, much more stable tumor regression than the MEK inhibitor itself, suggesting that this could be a promising combination. Okay, so I'm going to switch gears and just go through a couple other examples. That first example really excited us and told us that this could work. My lab looked at a BCR-ABL inhibitor, a Gleevec inhibitor resistant chronic myelogenous leukemia to try to understand how these cells were becoming resistant. We knew they weren't gatekeeper mutations. So we did MIBS profiling and we showed, in this case, the kinases on the right-hand side of the subcenter line are upregulated. We showed that Lin kinases was aberrantly upregulated. Along with it came ERK and MEK and a number of other kinases. One of the IKKs, part of the NF-κB pathway, was upregulated. And we can see this at the protein level as well as the activity level. So MIL-R, the resistant cells shown here. So this gave us important insight that these drug-resistant leukemias were now using Lin as a surrogate kinase. So we then turned to targeting Lin using dasatinib, or targeting pathways that we discovered like MEK or IKK downstream. And the results are shown here. Cell viability was drastically reduced with dasatinib, where inhibiting either of these branch pathways by themselves only gave a partial effect. Combination of these inhibitors gave a more sustained effect, much like dasatinib. And inhibitors like dasatinib are now considered alternatives for drug-resistant CML. But again, kinome profiling revealed how we should target kinases to prevent this adaptation. Okay, so I'll go through a few of these examples more quickly. We spent a lot of time studying pancreatic cancer with Channing Durr's lab here. Only recently have RAS inhibitors been developed. Previous to that, people were spending a lot of time using cytotoxics and still are, gemcitabine, FOLFIRINOX, or trying to target some of the kinases that are activated by RAS. And one particular example, we looked at what happens if we knock down RAS. This is Channing's lab. Angelina Vasiva was the lead author on this. And the MIBS profiling suggests that there was an upregulation of a kinase we hadn't expected, and that's known as MEK5, parallel path to the ERK5 pathway, and ERK1-2 pathway. And in conjunction with that was a strong upregulation of a kinase called ERK5. And so it suggested that maybe if we targeted this pathway in combination, there would be some benefit. And I will point out that in addition to RAS knockdown, ERK inhibition induced similar results. And the end of this long story was that basically RAS, the mutant protein, is driving this pathway, again, like we saw in the breast cancer example. If we inhibit one step further down with an ERK inhibitor, what we learned was that we upregulated through this loss of feedback, the EGF receptor, and then the MEK5 ERK5 pathway. So this was quite illuminating, was that there's a parallel pathway waiting to be activated that it could again phosphorylate our old friend MEK and then restore growth. And so what Angelina showed was that by co-targeting this pathway with a MEK inhibitor, a MEK5 inhibitor, and an ERK inhibitor, you could see much greater reduction in tumor growth. So again, the MIBS profiling alerted us to this idea that there was a separate pathway that could be co-targeted. Since then, there have been a number of publications, and again, I won't go through all these because of time, but just to show you that we've used this approach, in one case to define the specificity of CDK9 inhibitors and to demonstrate that CDK9 can also phosphorylate MYC. In another case to show that FOLFIRINOX, which is used for treatment of pancreatic cancer, upregulates a kinase called Jun kinase, and that by co-targeting that, we could reduce the cancer cell growth better. A very interesting example from our Minja Kettenbach's lab, we showed that we could compare kinome and phosphatase capture, where she made phosphatase inhibitor beads, and we could capture both kinases and phosphatases and learn something about how cell cycle was changing. Another study from Channing's lab, two more studies showed that by combined inhibition of kinases discovered in MAC, we could improve the apoptotic death of some of these. As I mentioned, a recent study came out to look at how we could study the effects of coronavirus, COVID, on different signaling pathways, and this revealed that the PI3 kinase mTOR pathway was becoming active, and that inhibitors of this could be potential antivirals. And then lastly, two papers that came out this summer that really used kinome profiling and phosphoproteome analysis to better understand, again, how ERK regulates the phosphoproteome in these KRAS mutant cancers. And that's shown a little bit more in this slide here. And we'll talk about how important it was that, number one, we knew that the ERK inhibitor was specific for its target, which allowed us to look at the phosphoproteome and discover which proteins were being phosphorylated by ERK specifically. Okay, so I'm going to switch gears a little bit again and talk about kinase inhibitor specificity and how we use this approach. So we and others have used this quite widely, and I should point out that Bernard Kuster's lab has done this at a large scale to look at many, many kinase inhibitors. We've focused more specifically on ones that are interested in our group. But the idea is that if a kinase inhibitor binds a kinase target tightly, when we apply this MIBS bead or kino bead in a profiling approach, any kinase that has an inhibitor already bound will pass through, whereas kinases that don't will bind. And by subtractive reasoning, we can discover which kinases prefer to bind this inhibitor and which don't. And so this has been a very powerful way for us to look at kinase inhibitor specificity and also to discover potentially new targets of kinases that we hadn't seen before. Now, the limitation is that not all kinases bind to these beads. We've tried to be as broad as possible, but we missed some. And that it requires tight binding of the drug to this kinase to begin with in order for this to work, so that if it's a poor kinase inhibitor, it may not work as effectively. And here's a couple examples that we'll talk about. We use this before we start any phosphoproteomic study with the kinase inhibitor, to determine the inhibitor specific. Is it inhibiting one kinase, 10 kinases, 50 kinases? We need to know that before we do these things. We also need to know, if we're going to study cell growth or other biology studies, how selective is this inhibitor? And then we also use it to look at, can we discover novel targets, novel drug targets for cancer biology, for malaria, for COVID, and other diseases, which we won't have time to go into, but we are working with a number of groups on this. So here's a really good example of an inhibitor that we were given from a group here at UNC, and they wanted to know how selective this kinase inhibitor was across the kinome. So again, to orient you, everything to the left of center line is displaced from the MIBS beads. Anything to the right increased binding to the MIBS bead. And basically, this experiment was done by treating a cell lysate or a cell with 10 nanomolar of the inhibitor, 100 nanomolar inhibitor, or 1,000 nanomolar of the inhibitor, and then looking for what we call dose-dependent inhibition. And you can see, for example, Axel up here in the upper right-hand corner shows beautiful dose-dependent displacement. So at the highest dose, you see that the kinase axle is being displaced very clearly. Others like MERTK don't show dose dependence, even at the lowest dose is completely displaced, saying that that's a very good target. But note, across the kinome, there's a lot of other kinases being inhibited, LATs over here, some of the CAM kinases. And you might say, well, these are just at the highest dose, but here's the kinase BMP2K that at 100 nanomolar, or AAK1 at 10 nanomolar, showing very strong inhibition. So that gave us a ton of information in terms of how did the kinome specificity tell us something about this inhibitor, and it's since been re-engineered to be more specific for its target. Okay, so very quickly, I'll go over a top little paper from our lab. We were screening kinase inhibitors, part of the SGC group here. known as PKIS1. We were looking for kinase inhibitors that would block gemcitabine-resistant cancer cells, pancreatic cancer cells. And we found a couple compounds that looked very very potent, that could potentially have some effect as an anti-cancer treatment, but we didn't know the target. So we did MIBS profiling, and to our surprise, we found a very strong hit on a kinase called CDK9. We found a strong hit on these kinases known as the ClpP kinases, and then some effects on a kinase called DYRK1. And again, to make long story short, what we did is we went through a number of studies testing other ClpP inhibitors, DRYK inhibitors, and showed that they had not the same effect on same cell growth as this compound. And then further analysis showed that CDK9 was the primary target based on kinase inhibitor studies, kinase modeling studies. So again, MIBS informed us that this drug that was effective against gemcitabine-resistant cancers was probably targeting this kinase CDK9. And there's a lot of studies and interest in CDK9 as an anti-cancer agent right now. Okay, another project we were working on, there were a number of papers that came out at the time when one of my graduate students, Ian McDonald, was in the lab, and they suggested that MELK was a key kinase in cancer biology. However, most of the studies were based on the selectivity of a kinase inhibitor, or the response to a kinase inhibitor known as OTS. And we had some questions about whether OTS was specific, and so there had been some publications suggesting that it was not specific at all. And so we got a hold of a couple of other kinase inhibitors that were designed to target MELK. And we did kinome profiling to determine if one or more of these could be used to examine the role of MELK in cancer biology. And so again, this is a hard slide to look at, but just to point out that the blue is OTS. And again, across the kinome, you can see that OTS at 1 micromolar is having huge effects on multiple kinases. The red bars are from this HDH compound, which showed effect on some kinases, but really no effect on MELK, and I'll show that in a second. Closer examination showed the best inhibitor of all was this 8A compound, which had very little effect on other kinases, but was very specific for MELK, and this was nicely reassuring. Again, here's OTS, here's the HDH, but as we already said, OTS inhibits many other kinases, even some of those in this panel here shown here. Okay, so I'm going to conclude this part and then go on to the next part. MIBS kinome profiling is incredibly powerful. We've used it to look at the biological response to MEK inhibition, to inhibitors designed to inhibit BCR-ABL, to RAS knockdown, ERK inhibition. It's told us a great deal about kinome reprogramming and kinome remodeling. And kinome remodeling has told us a great deal about how application of single kinase inhibitors to treat disease have its limitations, and that how nature quickly rebounds and can do so by transcriptional remodeling through proteins such as MYC. But fortunately, kinome reprogramming or kinome profiling has told us about how, if kinome reprogramming is occurring, how do we improve by using new combinatorial strategies? And I think this is what's exciting about this work, is that it gives us new insight into potential drug combinations. And as I mentioned, combining this with phosphoproteomics gives us a much better insight into what can be happening at the protein level. And then we can also use MIBS to better understand how selective our kinase inhibitors are we hitting the targets? And again, this is widely applicable to multiple systems, which we didn't have time to talk about today. All right, like I said, I'm going to shift gears one more time, and this will just kind of wrap things up. This has been a very exciting project for us, and it was rooted in the idea that initially that this compound known as ONC201 might have some important impact on kinases, and that's why we went looking for it. But just a little history. This compound was discovered in a screen for compounds that induce TRAIL, which is an causes cell death of cancer cells. And I have to point out this was all started by Wafik Al-Diri and his Penn State group. and work that they did in collaboration with the NCI. ONC201 has now shown enormous promise against multiple cancers, most recently this terrible brain cancer known as DIPG. And they've seen amazing shrinkage of tumors, leading to be a great deal of enthusiasm about this molecule. At the time we started working on this, the target for ONC201 was unknown. It had also been reported to be a potential dopamine receptor antagonist, but it wasn't clear from any studies that this was really right, nor was it trail inducing was consistent. And a pivotal paper from Yoshimi Greer and Stan Lipkowitz showed that mitochondria were the target for this compound. If they ablated mitochondria, the growth inhibitory effects were gone. And that's where we got involved. And I was working with a person from a company called Madera Therapeutics on a kinase-related project, and we became very interested in analogs to ONC201. So our goal was to test some of these analogs on cancers and to try and elucidate the mechanism of action. And so here's Ed Iwanowitz, founder of Madera Therapeutics, and these are some of the analogs that they built. And some of the first experiments in our lab, we just showed that compared to ONC201, all these new compounds, which we refer to as TR compounds, were more effective at inhibiting breast cancer cell growth. And so that was very exciting to us. We did some experiments. We found no evidence for dopamine receptor antagonism. And so we started looking at, well, what are these compounds actually targeting? And to do so, we had Madera make just like we did with the kinase inhibitors, an analog with an amine group on the end that we could use to couple to saffrose to go fishing for what might bind to this compound. And so I involved my younger brother in these experiments, and he helped make the affinity column. He coupled this compound TR80, which we knew inhibited cell growth, to the beads, and we took lysates from cancer cells or cancer cells plus the free ligand to ask what was binding and could we compete it off with this free ligand. And the results came back surprisingly interesting and clear. First of all, I will point out there's a lot of background, but one protein band showed up very clearly in the affinity column, and it was competed off with the free drug, suggesting that this was binding specifically to the column. So I used a razor blade to cut out the band, put it in the mass spectrometer, and it spit out, there were 8 peptides from this protein called ClpP. And I first told my brother that he screwed up, that it couldn't possibly be right, except the Greer paper told us it was mitochondria and targeting, and we knew that ClpP was in the mitochondria. And subsequent experiments from Paul showed that by Western blotting, ClpP was binding very specifically and it was competed off. I will point out that our paper was published, and a week later, Ishizawa and Andre published a nice paper in Cancer Cell confirming that the target for ONC201 was ClpP, and they did it by a completely different means than we did. So what is ClpP? Well, ClpP is part of this ClpXP protease system. It's much like the proteasome of the mitochondria, as shown here. It's a dual component protease. It has an ATPase known as ClpX, which unfolds proteins, funnels it into the barrel of ClpP, which is a tetradecamer, 2 dimers of 7 subunits. And then once the protein enters there, it's degraded into component peptides. So it's a very active protease that degrades unfolded proteins as they appear in the mitochondria. It's also conserved throughout nature, found in bacterial cells, C. elegans, and so on. So not surprisingly, people had been looking at ClpP inhibitors or even ClpP activators as antibacterial compounds. And there had been a few compounds that had been discovered by one of our collaborators, Dr. Hurry, and other inhibitors from Dr. Schimmer's lab had shown that there are compounds that will inhibit ClpP activity. But the role of ClpP in cancer biology was really not very well understood, and still isn't. But like I said, they were largely invested as antibacterial agents. So we teamed up with Walid's group, and he published a beautiful paper this last year. And our lab showed very specifically that these compounds activate the protease. And that told us not only did we find binding, but we activate. And this was published by one of my graduate students in 2022. And once we knew that, we gave some of the compounds to Walid, and he showed that by crystal structure, the compound binds very tightly into a very specific pocket in between these haptomers, opens up the pore of the protease, changes the conformation of the protease, and turns it into a hyperactive protease. No longer needing ClpX, it acts as a what we call a juggernaut protease, or as my colleague Ed Iwanowitz likes to call, Pac-Man set loose in the mitochondria. So we wanted to be sure that we had the right target, but given the discrepancy with the dopamine receptor and other trail-inducing studies. So my graduate student Emily, who's now at the Salk, showed again these TR compounds and other ONC-related compounds, very good inhibitors of breast cancer cell growth, as shown up here. But if we knocked out ClpP, and this was from cells that Yoshimi Greer sent us and our labs cells we developed, there were no effect of these compounds. We completely lost the effect. The dopamine receptor and other things were not important in this response. So our model at that time was that, okay, these compounds get into the mitochondria without any assistance. They activate this protease in the matrix. They cause mitochondrial proteolysis, loss of mitochondrial function, and then potentially we're starving these cancer cells of the ability to make ATP and can kill them in this way. Now we know that it's not as simple as that. In some cases, the cells don't die, they go into senescence, which I'll just mention at the end here. And it's not clear, but one thing that's been of great interest is that normal cells seem to avoid effects from these compounds. And so one of the main questions has been, why are cancer cells more selective? Is it because there's more ClpP protein in there? Possibly. We see that in some cancers, they've upregulated ClpP, and that might make them more sensitive to this. Are we targeting potential cancer cell metabolic vulnerability? We know that cancer cells have changed their metabolism. We have strong evidence that this may be the case. And as I mentioned, we don't always see strong evidence for apoptosis. There are papers now that have suggested ferroptosis is occurring, but in many cases, the breast cancer cells default into senescence. And then lastly, I will point out and just leave this very briefly with you. ONC201 initial studies suggested that tumors that had been exposed to ONC201 showed increased immune infiltration, that by targeting cancer cells with these compounds, we may make them more immune reactive. And that's something we're very interested in pursuing. Okay, so again, don't have time to go through all this, so I'm just going to very briefly summarize. We did a lot of omics, proteomics, transcriptomics, metabolomics, and we even did MIBS to try to understand how this works. MIBS has been very complicated. We're still trying to understand how the kinome is responding to this mitochondrial target. We learned a lot about what's changing at the transcript level, at the metabolite level, and the protein level. Not surprisingly, multiple proteins in the TCA cycle are downregulated, metabolites in the TCA cycle are downregulated. And if we look at mitochondrial respiration, we see that very clearly these compounds are affecting the ability of oxidative phosphorylation to work. And this is, again, note the low concentration, nanomolar concentrations of these compounds are very effective, where if we look at the knockout cells, there's no effect. Again, establishing ClpP as a target, and this was published in Emily's nice paper. So again, to summarize, through all our proteomics efforts, we've identified many cancer cell dependencies: pyrimidine biosynthesis, OXPHOS, cholesterol biosynthesis, mitochondrial DNA transcription translation, fatty acids. These compounds, through the activation of ClpP in the mitochondria, are having broad effects on multiple pathways, including proline biosynthesis, which is strongly upregulated in cancer cells, saying that, yeah, these are broad acting and could be very effective against cancer cells that have upregulated these pathways. We did find one pathway that was strongly upregulated, that is this integrated stress response. which said to us that the mitochondrial stress is being communicated to the nucleus, and that this result may also be important in some of the subsequent events, which in this case are cell senescence. So we're very actively investigating how senescence is being upregulated in these breast cancer cells, what pathways are involved in this, and then what it means to the treatment of these cells. And so I'll leave you with this model. And this is where we're going with a lot of this research. Again, the ClpP activators come into the mitochondria. They induce peptide degradation, widely inactivating oxidative phosphorylation and other things that the mitochondria need to survive. We know that as a result, we send out signals. In some cases, things like interferons possibly are going up. Increased levels of these cytokines known as SAS, which are associated with senescence. And that the release of these cytokines has been shown to increase the recruitment of immune cells. So our model being that through this mitochondrial stress, we're making cancer cells possibly more visible to this. And we've also done some cell surface proteomics that have shown that certain proteins have changed that may make these cells more reactive to natural killer cells. So one experiment I'll leave you with is that we've been treating these, and this is a TR57 compound, converting the tumor cells into senescent cells, then incubating them with natural killer cells, and asking if they've been pre-treated with TR compounds, can we now kill the cells more effectively with natural killer cells in vitro? And the answer seems to be yes. And we're working on what on the cell surface may be communicating to these cells to now come and get them and kill them. Okay, so I'll conclude this part here. I know I've gone over a little bit. I just wanted to say that this is a very exciting area of pharmacology. ClpP activators are now, a number of groups have dived into this. And, fortunately, the TR compounds are among the most potent that have yet to be discovered. ONC201 is moving forward in the clinic, as well as derivatives of ONC201 through a company called Chimerix. We know that multiple metabolic pathways are disturbed, and that we induce cell stress, and that this may be broadly effective at cancer for this reason. We're blocking both mitochondrial transcription and translation, and having an effect that may be difficult to reverse compared to other mitochondrial targeting agents. And then lastly, some of our data suggests that the targeting these cells, potentially making them senescent, may make them more susceptible to immune killing and so on. So this is many of the people here contributed to this work. I won't go through everybody. Some of the current members, Sabrina and Owen, Andre, one of my undergrads, and then my collaborator, Exxon, one of rotating students, Laura from South Africa, is working with us right now. Large group of other people, the proteomics, metabolomics groups, Yoshimi and Stan, of course, our collaborators, Waleed, my brother at Brooklyn Methodist, and then collaborators from the Dur Lab, and then my funding over here. But just to finish off, I want to just dedicate this talk to my father. He was actually the first student to study protein phosphorylation with Ed Krebs and Ed Fisher when they first discovered the kinases in the 1950s. He always told me that those guys would win the Nobel Prize. Well, he was a very instrumental part in helping them do so. And after working with them, he had his own highly successful career at Iowa State for over 39 years, where he studied protein kinases and protein phosphorylation. And he taught me everything I know about kinases and many other important things as well. So I just finished with that. Thank you for your attention. Happy to answer any questions.
Dr. Graves, thank you so much. And how touching to see that this work really does run in your blood. How extraordinary. I'm opening it up to the floor. If anybody has any questions now, you can unmute yourself and raise a hand if you'd like to get into the queue. But perhaps we'll start with, could you speak a bit, Dr. Graves, to some of the limitations to MIBS and kinome profiling?
Yeah, I think the biggest limitation is what are we putting on the beads? What are the inhibitors that we're using, and how broadly are we actually capturing the kinome? And what Gary Johnson's lab has shown is that some kinases in the AKT family and some other unusual kinases require unusual kinase inhibitors. So we've had to go back and re-engineer new inhibitors and redesign the MIBS beads to capture some of the less known kinases. And so it's in a way, it's kind of a crapshoot to see which ones we catch. And it also requires that the kinase inhibitor binds with a high affinity, which most do. As I pointed out, most bind at nanomolar affinity as compared to ATP. Um, but it is a binding assay. And so we can't infer everything about kinase changes just by binding, but by combining it with phosphoproteomic studies, we can get a great deal of insight. But that's a very relevant question. And again, how we apply these is really critical to understanding what the kinome changes are, like I mentioned before.
Thank you. Are there any further questions from our audience? Okay, a second one from me perhaps is, how do you see MIBS being used to discover the target for rare diseases going forward?
Yeah, I think one of the things that we're very excited about is, if we have a cancer where we don't know the driver and we want to know something more about the kinome profile and whether or not that's using MIBS can be used to fish out kinases that are less well known. And again, we've used this approach in some cases to look at some non-cancer samples, some malarial samples, some fungal samples, but also we're looking at other cancer-related or rare diseases to just ask very fundamental questions about what does the kinome look like? Are there certain ones that could be potentially targeted? Are there certain kinase inhibitor combinations that could be used that might make this a more effective treatment? I think that's the beauty. If we find one new potential driver, it gives us a target to go after, just like we did with the ONC201 studies. And it may be in some cases that it's not a kinase. It may be that it's another ATP binding protein that just happened to come along on the MIBS beads. And we do find some of those from time to time. So very good question.
There's a question in the chat from Lionel. Thank you so much for your talk. You mentioned two other techniques that you use, but focus more on the MIBS technique. Is the MIBS technique the, so to say, best of the others, or is it just more routinely used?
Well, it's the best for us because we have these beads and we can do this quite quickly. The KINActiv is a little bit more complicated, a little bit more expensive, but it works quite well to discover active kinases. So we're planning to do some direct comparisons. And then we almost always try to couple this with phosphoproteomics because that gives us a good insight into what the substrates are that are being changed. The problem being, again, that phosphoproteomics is fairly involved with the enrichment experiments we need to do and the isobaric tagging that we need to do. So it's a bit more expensive. It just adds more expense. So MIBS is pretty popular because my graduate students will do it and we can get answers quickly and less expensively maybe than the other ways. Good question. Like thank you for asking.
Yeah, sounds like a good answer. Thank you, Lionel. And then just, I think maybe finally, if there are no more questions in the chat, if you look back at your career, Lee, and you sort of think, how did I get to where I am now? What advice would you maybe give to younger researchers?
Keep your eyes open, keep working hard. It's ironic. I started working as a graduate student on protein degradation in bacteria and wonderful training that completely switched directions to go into eukaryotic protein kinases, which was quite challenging for me because I had to learn a whole new area, although my father was a kinologist, as you might say. But the funny thing is that our studies on ONC201 and the TR compounds has come full circle in that we've now come back to studying a protease that is in bacteria, and probably was involved in my earlier studies as a graduate student. And we've now discovered molecules that are activators. And I know there are groups looking at them as antibacterial. So you never know where science will take you. Just keep your eyes open and look for these unexplored avenues that you can pursue. And now, of course, we're asking, well, how does this mitochondrial protease affect the kinome? And how does that impact the development of senescence or an immune response. So all these things will come back to you, it seems like, in a long career, which makes it all the more interesting. Yeah, thank you for the question.
That's funny how that works. It's almost like a spiral. You revisiting places as you go on.
Yes, full circle.
Everybody, I've put links in the chat, both links to H3D Foundation and to Dr. Graves's work and lab. And also there's an opportunity, as always, to give feedback. Often our speakers love the feedback that you give. So please do, if you have a chance, you can send some comments through the Google form. And so that just leaves it to me to end our session today and to thank Dr. Graves. Thank you so much for your time and expertise and for sharing your knowledge so generously with us. I know it is really always appreciated. Thanks to all of you for tuning in. I think our next one is not next week, but check your inboxes and you'll see when it's time to register. Thanks so much, everybody.
Thank you, Nicola. Really appreciate this opportunity.
Wonderful. Bye.
Bye-bye now.
Bye-bye.